My name is Barbara Melchert and I'm from the Department of Molecular Pharmacology of the University of Groningen. Today's date is March 30th, 2020, and this is a recorded class for the course Pathology and Immunology for first-year pharmacy students. I originally gave this class on March 6th and I re-recorded it for a complete set of online lectures. So this class is about B and T cell development, because I told you about how B and T cells recognize antigen and can get activated. But how do they actually know to respond to things that are a danger? How do they recognize self and non-self? And that is something that they learn when they grow up. So B cells know to attack invaders outside of cells, and T cells know how to attack invaders that are inside cells, and they also know exactly what is self and non-self. And that happens in the generative lymphoid organs. So both B and T cells develop in bone marrow, and B cells stay in bone marrow and then develop into mature B lymphocytes. And T cells, after being born in the bone marrow, move as progenitors to the thymus, and there they develop into fully functional mature T lymphocytes. Then these mature cells go to the lymph nodes and start circulating in those lymph nodes. So they go to the peripheral lymphoid organs. And what they learn during growing up in either the bone marrow or the thymus is how they should behave, what they should do, what is self and non-self, and if they cannot make the distinction, then they're being deleted. So they are trained not to recognize self. So when they grow up in the bone marrow or the thymus, if a lymphocyte that is present there and is growing up and is starting to express B or T cells, if those B or T cells bind self-antigen, then they're going to die by apoptosis. If the antigens that grow up there do not recognize the tissue that they're in, if they don't get activated by that, then they're allowed to continue to mature further and become fully functional B or T cells. So T cells not only have to be able to not recognize self, but they also have to make a choice whether they want to become a CD4 or a CD8 positive T cells. So the progenitor cells from the bone marrow move to the thymus, and these pro-T cells then develop further. And what happens is they first develop both CD4 and CD8. So they're starting to express the CD4 protein and the CD8 protein. And then they're going to test, do I recognize MHC class 1 or do I recognize MHC class 2? Because the CD4 on CD4 positive T cells specifically recognizes MHC class 2, and the CD8 on CD8 positive T cells specifically recognizes MHC class 1. And if they recognize MHC class 1, they continue to be a CD8 positive T cell. If they recognize CD4, then they become a CD4 positive. If they recognize MHC class 2, then they become a CD4 positive T cells, but only if they do not recognize self. If they recognize self a little bit, then they're also sometimes allowed to become a regulatory T cell. A regulatory T cell is a T cell that downregulates T cell responses and actually prevents autoimmunity, so a response against self, because it can downregulate the response of other T cells. So if this T cell recognizes self, it can tell other T cells, don't respond to this, this is self, please keep quiet. And how does a regulatory T cell do this? So here we have the antigen presenting cell that presents antigen in the context of MHC class II, that is recognized by a CD4 positive T cell. And this CD4 positive T cell can get activated, but if there is a regulatory T cell that recognizes this antigen as self, it can tell this T cell, don't do anything, don't have to respond, just keep quiet, this is self. So in the thymus, T cells basically have three choices. Either they recognize self and they become a regulatory T cell, or they recognize MHC class I and become a CD8 positive T cell, or they recognize MHC class II and they become a CD4 positive T cell. And then when they move out of these generative lymphoid organs and go to the periphery, to the lymph nodes, and then recognize something, then they become activated, and then you get effector immune responses. For a CD8 positive T cell, that means that it becomes a cytotoxic T lymphocyte. And for a CD4 positive T cell, it means that it can choose what it wants to be based on signals it gets from the dendritic cell. It can become a Th1, a Th2, or a Th17 cell. And that will be explained in more detail in other classes. So what happens then is that you get immune responses. And in short, this means that here we have the lymphocytes that get activated either by extracellular antigens or antigens presenting by other cells. Then they get activated, they start proliferating, so they start to divide. You get clonal expansion, which means that every clone, every T cell or B cell that recognizes specific antigen starts to divide and starts to make brothers and sisters that recognize the exact same antigen. Then they differentiate into an effector cell, so that could either be a plasma cell that produces antibodies or a effector T lymphocyte, so a Th1, 2, or 17 cell, or a cytotoxic T lymphocyte. Then they start to eliminate the antigen, and then when the antigen is eliminated, then all those excess cells will die off, and a few will survive as memory cells. And we'll talk about this more in the next couple of videos.